Neuropsychologists study split brain patients to probe the unique functions of the left and right brain hemispheres. In other words, to study lateralization, and to also investigate the nature of communication between these regions. Primarily speaking, information from one side of the body is processed within the opposite half of the brain. In addition, each hemisphere contralaterally directs body movements. These areas also have different cognitive strengths. The left side is typically associated with the control of language and speech, whereas the right plays a large role in processing visual spatial information, like judging the spatial arrangements of dials on a machine. Normally, collections of neurons' axons, referred to as nerve fiber bundles, transfer information between these hemispheres. One of the largest of such tracts is the corpus callosum. However, this interhemispheric communication is interrupted in split brain patients whose corpora callosa have been surgically severed, a treatment sometimes used to reduce the uncontrollable neural activity characteristic of epilepsy from spreading throughout the entire brain. Using modernizations of psychologist Michael Gazzaniga's techniques, this video demonstrates how to test split brain patients and assess their cognitive abilities, specifically speech production, and illustrates data collection and analysis methods. In this experiment, patients are shown images of everyday objects and asked to verbalize the name of each item. To achieve lateralization, patients are instructed to focus on a cross symbol in the center of a computer screen and told to remain fixated on this shape for the duration of the experiment. Here, the cross serves as a reference point next to which visual stimuli can be shown on either the right or the left. If an image is presented on the right side of the screen, it falls into the right visual field, which, perhaps counterintuitively, is processed by the left portions of both eyes. These regions then project the observed image to the left hemisphere of the brain, where it's identified. Thus, functions of the left brain hemisphere can be assessed by showing images in the right visual field. Similarly, a stimulus presented to the left of the cross on screen in the left visual field can be used to evaluate the roles of the right hemisphere. During the naming objects task, a total of 50 drawings, like that of a chicken, appear one at a time on a random side of the monitor, either the right or the left. Pictures are presented for less than 150 milliseconds. As this is not enough time for a patient to move their eyes to reposition the image, it ensures that only the brain hemisphere being tested sees the stimulus. After the image disappears, the patient must identify it aloud, which serves as a measure of the lateralization of verbal linguistic capability. Here, the dependent variable is the percentage of images shown in the left and right visual fields that the patient is able to name. In other words, the accuracy of verbal identification. Based on the previous work of Gazzaniga and others, it is expected that patients will be able to name images presented in the right visual field with high accuracy, as this information is seen by the left hemisphere, the region capable of controlling speech. However, patients will be unable to verbally identify pictures shown in the left visual field, as this information is handled by the right brain hemisphere, which is incapable of generating language, and in split-brain patients cannot communicate with the speech-capable left side. If the image can't be named, referred to as anomia, a drawing task is performed, which serves as a non-linguistic measure of stimulus knowledge. Here, patients must create a picture of the image they were shown using the hand on the ipsilateral, or same side as the tested visual field. Thus, if patients can't verbally identify an object presented on the left of the screen, they should draw it with their left hand. In this instance, the dependent variable is the percentage of images shown in the left and right visual fields that are accurately drawn. It's expected that patients unable to name pictures shown on the left of the monitor will still be able to draw them, using their left hand with high accuracy. This is due to the fact that the right hemisphere which controls the left arm and hand, also processes information from the left visual field. Thus, no communication is needed between the hemispheres to complete this task. Prior to beginning the experiment, review patients' MRI data to determine which nerve fiber bundles they're lacking. For the purpose of this demonstration, a patient in whom the entire corpus callosum has been severed is tested, and their data will be compared to those collected from control participants. Greet the patient when they arrive and inform them of the research procedures. In addition, assure that they have signed all appropriate consent forms. Then proceed to place their chin comfortably in a chin rest so that their eyes are positioned approximately 22 inches from the screen. With the small cross displayed in the center of the screen, emphasize to the patient that they must remain fixated on this symbol, even as images flash to the left or right of it. Proceed by showing them 50 images, each of which are presented for 150 milliseconds in a random order and evenly divided between sides. After every presentation, instruct the patient to identify the object out loud.
Apple.
Record all of their responses. If the patient cannot name the visual stimulus, ask them to draw it with the hand on the same side as the visual field in which the picture was shown. This constitutes the drawing objects task. Make sure that the patient does not look at their hand as they're drawing, in order to maintain the initial isolation of the stimulus to one brain hemisphere. To confirm that the patient knows the name of the stimulus when it is simultaneously presented to both fields of view, have them look down at their completed drawing and verbally identify the object it represents.
Is it a broom?
Again, record all of the patient's responses. To analyze the data, first collect the percentage of correct verbal responses across patients for stimuli presented to the left and right visual fields. Proceed by separately compiling the percentage of accurate verbal responses for each control participant's left and right locations. To identify any deficits in patient behavior, compare control and patient data using a repeated measures analysis of variance test. Repeat the analysis for all data collected from the drawing test. Notice that while patients are typically unable to name stimuli presented to the left visual field, they can draw them with their left hand with a high degree of accuracy. This demonstrates a dissociation between a patient's ability to recognize and verbally name an object. Now that you know how to test the functions of the left and right hemispheres of split-brain patients with visual stimuli, let's see how researchers explore and apply lateralization in other contexts. You've learned that surgical separation of the two hemispheres is often used to treat patients with epilepsy, which is characterized by seizures. As a result, many neuroscientists are looking at whether the timing of this disconnection, whether the corpus callosum is severed during childhood or adulthood, has any effect on a patient's cognitive functions. Importantly, such work has demonstrated that compared to adults, children experience fewer or less severe cognitive effects following the disconnection of the brain hemispheres, suggesting that young brains demonstrate a great degree of plasticity. Up until now, we've focused on the corpus callosum as the major connection between the left and right hemispheres. However, other nerve fiber bundles allow for communication between the sides of the brain. Among them is the anterior commissure, which has been implicated in the transfer of sensory information, like that pertaining to sight or smell. Thus, some researchers are looking at how the disconnection of one or more of these bundles, with or without severance of the corpus callosum, affects patient behavior. You've just watched Jove's video on testing split-brain patients using visual stimuli. By now, you should understand how to present images to the two visual fields, and collect and interpret data relating to the abilities of the left and right brain hemispheres. You should also know how data from split brain patients is being used to develop better treatments for epilepsy, and understand the roles of different nerve fiber bundles in the brain. Thanks for watching.